Okay, welcome everyone. Thank you all for being here. I'm sure people will continue to roll in over the next several minutes, but let's get started. I'll start by introducing myself. My name is Tyler Steven. I am the technical program manager for Current, and I will be getting us started for today's Third Coast Water Seminar, which is featuring the work of Dr. Ying Li, who will be talking about water membrane design and innovation. Really quickly, I want to thank the Third Coast Water Seminar committee, who is in charge of selecting the speakers for this series. It's a range of researchers from academic institutions across the Great Lakes region who are focused on selecting speakers who are giving talks around the general topic areas of interest to Great Lakes Renew, which is transforming waste into wealth. So this seminar series is developed as part of the Regional Innovation Engine Great Lakes Renew, which is focused on water innovations in the Great Lakes region. And this innovation engine is backed by the NSF and anchored by Current. Just a little bit of background on Current and Great Lakes Renew. Current is a water innovation hub based in Chicago. We were founded in 2016 with the goal to catalyze the growth of the circular blue economy, accelerate water innovation by convening and connecting various stakeholders, including researchers, entrepreneurs, and communities throughout the Great Lakes region, as well as to solve pressing water challenges while driving economic growth and opportunity, protecting human and environmental health, as well as protecting our greatest national security resource, which is our fresh water. Current is the quarterbacking organization behind Great Lakes Renew, which, as I mentioned, is a regional water innovation engine that is backed by the NSF. We started this work in January of 2024. And through the NSF, we have up to $160 million of funding over the course of 10 years. And the driving factors behind Great Lakes Renew is similar to like Third Coast Water Seminar Series and Current in general, which is transforming waste into wealth, specifically focusing on the selective separation and recovery of energy, nutrients, critical elements, and water from our drinking water, our wastewater, our surface water, as well as the selective separation and destruction of harmful contaminants such as PFAS. Through Great Lakes Renew, we have three key objectives, which is to spur the circular blue economy transition, focusing on really that circularity aspect of recovering what we can from waste and turning it into value and recovering valuable resources and turning it into economic opportunity. Really focusing on investing in the invention and commercialization of selective separation technologies from bench to scale, and attracting and retaining water-intensive industries in the Great Lakes region. Here are some of the key research and development areas that we are focused on currently. Throughout that general selective separation and recovery, we are focused on four key challenge areas, which is materials platforms, sensor platforms, process technologies, and subsystem testbeds. So this includes work such as PFAS detection centers out of the University of Chicago, as well as materials work such as reusable sponges for nutrients recovery out of Northwestern Purdue. That was really quick. If you would like to learn more, we encourage you all to follow Current and Great Lakes Renew on socials. If you scan that QR code, that will take you to the Current website. But without further ado, what you all came here for, I'm going to pass it over to Dr. Yupeng Zhang from Argonne National Laboratory, who will be introducing today's speaker.
Thank you, Tyler. My name is Yupeng Zhang. I'm a senior principal material scientist and a group leader at Argonne National Laboratory. I'm working on selective membrane design and synthesis scale-up for water treatment and critical mineral extraction. I serve on the committee of the Third Coast Water Seminar Series. It is my great pleasure to introduce today's speaker, Dr. Ying Li at the University of Wisconsin-Madison. Dr. Li joined the University of Wisconsin-Madison in 2022 as an associate professor of mechanical engineering. His current research interests, including multi-scale modeling, computational material design, mechanics and physics of polymers, and machine learning accelerated polymer design. Dr. Li's achievements in research have been widely recognized by fellowships and awards, including National Science Foundation Career Award in 2021, Air Force Young Investigator Award, and the 3M Non-Tenured Faculty Award in 2020, ASME Heisman Young Investigator Award in 2019, NSF CSE Research Initiative Award in 2018, and multiple Best Paper Awards from major conferences. Today, Dr. Li will present his research related to water membrane design and innovation. So with that, Dr. Li, the floor is yours.
Yeah, thank you. Thank you very much, Yuepeng, Taylor, and the current team. So thank you so much for having me here and offering this great opportunity. Let me share my screen. And, okay. So can you see my screen now?
Yes, you are good.
Okay, that's great. All right, so thank you so much, and good afternoon, everyone. So I'm really very excited to talk about our recent work, focusing on the water membrane design and innovation. And I let us add a small title here, since I'm a modeler, I'm a computational person instead of experimentalist. So this is my own perspective from molecular simulation point of view, as well as how we use machine learning to accelerate the polymer membrane design process. And the work has been mainly supported by NAUI. So it's a National Alliance for Water Innovation. It's a over $100 million DOE-funded clean water energy hub, as well as National Science Foundation and BSF, as well as Wisconsin Alumni Research Foundation. And a lot of research has been ongoing through a tremendous collaboration with Professor Manny Alamelik's group at Rice University and my postdoc, as well as Jeff McCutcheon's group at UConn and Eric Hoch's group at UCLA. So I want to start a quick introduction about the background and the motivation. So why we care about this membrane design and innovation, and then I will move on. As I mentioned earlier, so I will talk about two major parts. So one is focusing using molecular dynamic simulation, understand the membrane transport process. And the other one is how we use machine learning to guide this membrane design. And then I will highlight a few knowledge gaps and the research needs and conclude there. So I always like to using this water stress map in the United States. So a lot of you may not familiar with like the pressure of the clean water supply issue. So even though we think most of us located in the Great Lake region, right? We thought, oh, we have the Lake Michigan, one of the largest lake in the country, and we may not have this clean water supply issue, but which is actually not true, because surrounding the Lake Michigan, like Wisconsin, Illinois, and Michigan, so we do have the water stress, and although a little bit less than California and many other Western states, but we still have this water supply or clean water issue. And to enrich our water supply, so reverse osmosis or desalination technique is currently the most energy efficient technology people has been using. So the basic idea is actually very simple. So you have this reverse osmosis module, and you feed in the seawater or the wastewater, and you want to extract the pure clean water after the process. And the basic idea, behind this reverse osmosis module is actually you have a membrane, and this membrane play as the barrier, selectively allow the water going through while reject or the block the ions and the other contaminant within the wastewater. So this is a very simple idea. And the whole thing is not very complicated at all. So you can imagine, so you place your salmon permeable membrane over here, and with the support. And when the untreated water or the wastewater and showing up here, and you apply the pressure and press them going through this particular membrane, and this membrane as the selective layer only allows the water molecule going through. Well, reject the other contaminant. Okay, so that's the way we are able to purify the water and enrich our water supply issue. But this membrane material is not easy actually to discover or design. So a lot of you may not familiar how we cover or discovered this semi-permeable polymer membrane. So we actually start many years ago, actually back to 1960s. So people start testing some of the classical materials to see whether they are good enough, okay, to allow the water going through while reject the salt ions or some others. So it takes many years effort. So this is actually a very complicated roadmap. People tested many other materials during this process. For example, polysulfone support to some other cross-linked, polyamides, and following some other polymer innovation like some polyether, polyurethane design, and many other. And until, okay, so 1980s, and they discovered there is a very interesting recipe. So it's based on this MPD monomer and reacted with the TMC monomer. And these two, once they meet each other at the interface, during the polymerization process, you can come up this fully cross-linked aromatic polyamide. And this one has a very nice balance of reasonable high water permeability. Well, they have fantastic salt rejections, over 99%. So in this way, so we are able to efficiently extract water molecules from this seawater or the wastewater and will block majority of the contaminant, right? So that's the basic idea. But this is actually not enough because we are always looking for a better options. Although the water permeability is good enough, but there is more room to play. In particular, once you want to increase the energy efficiency, right, during this water treatment process. So that's the reason over the years, from 1980s up to now, and people start exploring, so what kind of the new strategies or the molecular design strategies we can bring in to improve them. So one way is trying to play with the different molecular structure of the monomer when you synthesize or design this polyamide membrane. For example, MPD as a standard benchmark reference, people start playing with some other amine molecules and trying to introduce some weird and contorted, even this kinked structure. And what they found out is actually if you are able to replace the MPD monomer by using these weird geometries of the amine molecule, you actually can open the internal fraction-free volume of the membrane. So they will allow you to achieve a higher water permeability while maintain a similar or comparable salt rejection. So this is known as a classical permeability selectivity trade-off for the polymer desalination membrane, so which limited the efficiency and we are facing. So that's typical the upper bound we're trying to plot with if you use the permeability versus the selectivity, okay? So we currently limited by the MPD-TMC recipe for the polyamide, like I mentioned earlier. Well, we're trying to push the upper bound to achieve a better membrane, which can offer us a higher water permeability and eventually better selectivity. So you can increase the energy efficiency during the design process. But this is not the end. And in particular, so we still have a lot of fundamental knowledge gaps during this reverse osmosis, or generally speaking, so this water membrane design problem. And nowadays, if you don't know the question, you actually can ask the ChatGPT, so which summarize very nicely. So this is 5 items. They quickly give it to me. So one is talking about the molecular pathways for the water transport. So how the water really going through a very dense, a heterogeneous molecular structure of this polymer membrane during the RO process. We don't really have a clear picture because the scale is down to the nano or sub-nanometer. We can now use our current experimental technique to visualize the whole process. So this is a particular reason why molecular simulation becomes very powerful to understand this nanoscale phenomena. And of course, the ion transport and selectivity. So this is particularly important. Nowadays, like we want to do the lithium extractions, critical minerals, and what is the governing factor influencing the rejection or the transport mechanism of the ion going through these polymer membranes. And of course, another very important aspect I already mentioned, so that is how the membrane chemistry and the structure influence their permeability selectivity, right? So how can we change the molecular structures, the cross-linking degrees, as well as the nanoscale inhomogeneities will influence the permeability selectivity, upper bound. And as well as some others like the external driving force and how we use the molecular level computation and the experimental insights to further improve these membranes. Okay, so these are just the 5 questions that ChatGPT quickly pop up, gave it to me. But for the limited time, so in today's webinar, so I will mainly talk about the 2 questions we are trying to address. So the first one is, what's the molecular pathway for the water transport? So this is particularly important. If we don't understand how the water going through the membrane, and we don't know how to really engineering the membrane to achieve better water permeability, so not to mention breaking the permeability selectivity upper bound. And the other is definitely there are so many chemistry design we can play with these membranes. People already spent over 40 years come up with the current recipe. So can we do better? And in particular with nowadays with artificial intelligence and machine learning techniques, can we make it better? So these are the 2 questions I'm trying to address during today's webinar. So the first one I will show you is how we use molecular dynamic simulation to quantify and understand the water transport mechanism across these polyamide membranes. We have been using over 40 years already. So to understand this, so first of all, we need to make sure. So we are doing the molecular dynamic simulation are really representing the real world polymer membrane rather than we are just playing with a toy model. So the simulation results or the insight you gain are not applicable to the realistic RO system. So that's the reason why we did this very careful benchmark study. So what we do is we take the same MPD monomer and we take the same TMC monomer, what experimentalists used in their synthesis, and then we put the mixture together, and then we cross-link them following the similar strategy as the experiments. You will have a dry membrane being formed at the beginning, and then you can place this cross-linked membrane and sandwiched between the feed of the water and the permeant side of the water, and you can apply the graphene pistons on both sides to create a pressure difference. So for instance, from the feed, so we can apply a much higher pressure to mimic the driving force during the reverse osmosis process, while on the permeant side, you can maintain the standard atmosphere pressure. So in this way, so the pressure difference will be able to drive the water molecule going through the membrane. So we will be able to track exactly how individual water molecules going through the membrane during the RO process. And to make sure, so we are really computationally generating the polyamide membrane in the realistic manner. So we start with our initial MPD TMC monomer. And so we monitor what kind of the molecular species being formed after this cross-linking process. So we consider how many TMC molecules are being reacted with MPD molecule. For example, TMC1 means only 1 reacted, and 2 means 2 reacted, 3 means 3 reacted, and similar. MPD can be just 1 reacted or 2 reacted. And we also monitor some other signatures like oxygen species and the nitrogen species within the polyamide membrane. And traditionally, the experimentalists only use the oxygen over nitrogen ratio, trying to back out the corresponding cross-linking degree. But we break it down, and we want to make sure we are generating a more realistic experimental relevant polymer membrane from our molecular simulation. So we use 2 of the well-known experimental results. So one from Andrew Livingston's group from UK, which did a very beautiful characterizations on the different molecular species as well. And the other from NIST, Christopher Stafford, which again, so they really experimentally characterized the different species like I mentioned here. And so we do one-to-one comparison. So as you can see, overall, our computationally generated polyimide membrane has a very similar molecular species, oxygen species, nitrogen species, ON ratio, and the cross-linking degree as experimental sample. So this gives us the confidence that so we are able to generating a experimentally realistic polyimide membrane for our molecular dynamic simulation. And then, so once you have the initial model, so you can start assigning the force field. So in the field or in the past, so people just randomly select whatever force field they have. So you can start running the molecular simulation and generating the results. But as a computational person, we know that doesn't work because a lot of force fields, they are just a general purpose. They are not specifically designed for the polyimide membrane, not to mention, so they will automatically give you the correct answer. So that's the reason why we chose 6 representative force field from PCFF, which is a polymer consistent force field, CVFF, Swiss Paragen force field, general charm force field, general amber, and the drilling force field. And then we can use our molecular simulation to quantify how robust or effective they are of the different force field to give you the membrane's property. So the first one we did is actually just stretching the dry membrane so we can get the stress strain curve of this polyimide, and then we can monitor what is the Young's modulus, okay, and from the initial linear elastic deformation regime. And our simulated membrane, majority of them actually give pretty decent predictions on the Young's modulus of the polyimide. In particular, the 3 of them are highlighted, and the dashed line are what experimentally has been measured, okay, as you can see. So they are pretty close to each other, nearly about 3 gigapascal. And we also quantify the dry membrane density, and the dry membrane density has different reported value depending on how you experimentally make these membranes in the lab. And but overall, we do see a similar consistency between our MD simulation versus what experimentally has been reported. And the most important thing, since the membrane allows the water molecule going through, will reject, ions or the other contaminant in the water. So we monitor the void or the pore size distribution of the membrane, in particular the primary pore, which decided how the water molecule going through. So the dashed line again is from very expensive pulse experiments, while the solid line are from our molecular dynamics simulation. So we see overall they have pretty good consistency. In particular, when you look at the average of the pore diameter as well as the porosity of the membrane, so the variety of the force field, so we use actually, so they can correctly, reproduce the average pore diameter as well as the porosity of the membrane, which is about 30%. So this gives us the confidence, right? So if you build the membrane correctly with the right molecular structure, and also you can apply the right force field, so you are able to recover the dry membrane property. And then we can move on to study the hydrated membrane. So as I mentioned earlier, so we can add in the water molecule, place the whole system under the piston so you can drive the membrane. And to quantify, the hydrated membrane, their swelling, and as well as the water permeability. So let's look at the swelling of these membranes. So again, so as you can see, after the hydration, so the membrane swells, they normally has over 1.2 swelling ratio, and which is very closely to mimic what, again, experimentalists has been reported, represented by this dashed line. Only very few of them, like general amber force field and the drilling, so they are not able to, reach the well-hydrated status. And the next one we also monitor is actually the water diffusivity within the membrane. So this is actually before you apply the pressure gradient and to look at the reverse osmosis process. And what we see here is actually very interesting. So we look at the 3 important water diffusivity. One is the bulk water diffusivity. So that is the water not within the membrane. And the other is the water diffusivity within the membrane, in particular in the confined region, because the membrane has the pore and the water molecule are being confined in this nanoscopic voids. And we look at their short time diffusivity as well as the long time diffusivity through the mean square displacement calculation and versus the broad range of the temperature. And we are also actually able to use the experimental data from the UK group, it's Empirical College London, who spent 2 years using neutron scattering back out the water diffusivity within the membrane and outside of the membrane. And as you can see, so our molecular simulation again can pretty reasonably predict these water molecule diffusivities, no matter they are outside of the membrane or within the membrane. Okay, so this is a pretty exciting point, as a pure simulation model can predict what experimentalist has been measured directly, and they are one-to-one comparison. So this is a very exciting point. And then we move on regarding the water transport to look at how much water we really can squeeze through, after applying a pressure gradient. And here's what we did. So considering the typical, the pressure, the reverse osmosis process able to apply from 0.3 to 1.5 kilobar. So we apply the pressure gradient. So we clearly see the water molecule coming out from the polyamide membranes from the feed side and from this water flux. So we actually can quantify how much water molecule we have, and correspondingly what's the water flux we have by dividing the membrane surface areas. And this is overall what we get again. So regarding the different force field choices we have, so as you can see, so with the pressure increase, so we do see the water flux is also linearly increase, not surprising, but the different force field indeed give us very different water flux. And in particular, so the first three force field give us a pretty reasonable water flux or permeability prediction in comparison with what experimentalist has been measured, while the last three, they didn't. And in the meantime, we do see a little bit of membrane compaction, because once you apply the pressure high enough, you really compress the membrane quite a bit. So we do see some clear membrane compaction behavior happening once the pressure gradient is increasing. And so to understand really how the water molecule going through these membranes, so we, from our molecular dynamic simulation, so we extract the membrane's pore structure. So these are the regions the water molecule may able to occupy and eventually going through during the reverse osmosis process. So we have three major categories of the pore within the membrane. So one is the relatively isolated pore, so represented by the red color. So they are just like isolated pore within the membrane, water molecule difficult to get in, difficult to get out. We also have some semi-permeable pore, so represented by the blue color, okay? So which means they are just halfway or just a little bit portion of the membrane, they don't cross the whole membrane. And the majority of them we discovered are these green color, so which represent the water channels across or span the whole membrane thickness, okay? So as you can see, so the force field we use actually greatly influence the microstructure of the membrane, in particular, the three force field I mentioned earlier, which gave us pretty good water permeability or permanence predictions, are having this green pore channel. So because they allow the water molecules easily going through the membrane, so we can predict experimentally relevant water permeability from this molecular simulation. While the other force field, so they mostly have this isolated void, which does not allow water molecule going through the membrane. So we don't have the correct water permeability from the simulation. So this gives us a very important lesson. The water molecule really going through the membrane through the channels. These channels are really spanning the thickness direction of the polyamide membrane. And we also look at the water transport at a relatively higher pressure. In particular, we look at how the channels are being formed during the whole process. In particular, so we are able to get some experimental values on the accumulated pore size distribution to compare with our molecular simulation. So as you can see, so what experimentally measured about the pore size distribution very closely from our MD predictions. So this is again give us the confidence the water transport characteristics as well as the pore has been correctly predicted by our molecular simulation so far. And here's a quick summary regarding the dry membrane properties, the hydrated membrane property, and what kind of the membrane properties you want to predict. So we provide a very detailed recipe. Which force field combination will give you good or reasonable prediction, or which one they don't at all. So the red one in particular, I want to point it out, you cannot just randomly assume they will work and use them to do this molecular simulation. Otherwise, you will get a completely wrong answer. And in particular, like I mentioned, so the water transport across this polyamide membrane is governed by these water channels. And in this particular field, we call it a pore flow mechanism. So this is very significant different from what classically has been assumed by water dissolving in the membrane and diffuse through, which is so-called a solution diffusion mechanism. So what we discovered is that water transport through the polyamide membrane is through a channel, and this channel is forming by this nanoscopic pore size, and which governs, how fast the water molecule going through. So apparently, the larger of the channel, the less tortuosity, the water permeability will be higher, right? So it's a very simple idea. And so this is another work we actually published with Professor Manning Alamanni's group. So we do quantify these detailed characteristics. In particular, so we see again a very linear water flux versus the pressure you apply. Well, the classical solution diffusion model give a nonlinear relation, which is never been observed from our MD simulations. And the other is that there are very clear water pressure gradient within the membrane, okay? So from the initial fade to the permanent side, so we see a very clear pressure gradient within the membrane, which provided the driving force, drive the water molecule going through these membranes, as well as the water concentration within the membrane is remain, constant. So that, disapproves the solution diffusion mechanism, which assume there is a concentration gradient for the water, which drives them going through the membrane. And of course, like I mentioned, so we can monitor the molecular trajectories of the water molecule during this transport process. So we see a very clear water cluster, not single file, a water cluster. They going through the membrane very quickly during the simulation. So again, showcase the pore flow transport mechanism for the water molecule. And here's a quick summary about what we see from these molecular simulations is water permeation happens through this free volume region within the membrane. So that's the reason why the microstructure of the pore engineering of the polyamide membrane play a very significant role. And we see the water going through the membrane either through some jumping mechanism, if you have relatively isolated voids, or the smoothing mechanism, they diffuse continuously through the connected channel. So the smoothing mechanism actually plays a major role. Otherwise, you don't get a reasonable water permeability. And at high pressure, the membrane has a compaction, okay? So this is a very important implication for the molecular simulation because once you have a validated computational framework with a good force field, so we really can predict a lot of important properties as well as the phenomena, okay? And in particular, so the membrane spanning pore, so the green channel I have highlighted in my previous slides, is crucial to achieving the experimental agreement regarding the water permeability. So this is again showcase how important the microstructure of the membrane plays a role, okay? And so next part, so I will move on to our machine learning study. So this is a very recent work we have been developing. So how we can use machine learning to further, design the polyamide membranes in addition to the traditional try and error experimental testing. As we know, or as we mentioned earlier, so the membranes are being formed by the MPD and the TMC monomer. So this is the classical MPD recipe. And in addition to the MPD-based amine molecule, we have many other amine molecules you can play with. Here I just show quite a few of them, which has been reported in the literature already. And some others even try some more complicated, amine molecules in the past, and they give you very different, salt rejection as well as the water permeability. And these monomers, they can, dramatically change the cross-linking degree as well, and also change the membrane homogeneity as well as the microstructure, the pore. And all of them together is playing a very important role if you really want to engineering your polymer membranes to break down the permeability selectivity trade-off, or you tailor their selectivity regarding the different ions or even the PFAS, rejections. So the design space is really huge. And in particular, if you look at the polymer design, we published a paper just earlier last year. So we talk about the polyamide alone, you already has trillions of choices. Compare with the existing polymer, we know just a few thousands of them. So they cover a much larger, much underexplored design space we never studied or we never imagined before. So this is really limited, our current capability to study all of them and to eventually identify what is the optimal recipe for our polyamide membranes. So we want to achieve the certain water permeability, the ion rejection, or the ion selectivity. To resolve this issue, so if you can ask ChatGPT, which will tell you, you will need autonomous labs on this polymer synthesis design and characterization. So hopefully they will, by end of the day, they will give you the best recipe. So this particular membrane give you the water permeability like this, ion rejection like this, and many others, right? But we know that this is not happening yet, although from Hollywood movie, probably we saw some of them. This is not happening yet. So behind these robotic platforms or the self-driving labs, what we really need is the machine learning modules to drive these robotic arms to make the decision, to synthesize the membrane, and to get the selection of the next candidate for the experimental testing or the characterization. To do that, what we need is actually, we need these two major modules. So one is the forward predictive machine learning model, which tells you or establishes the linkage between the synthesis of the polymer, their structure, as well as the property, okay? And for this particular one, you need a data repository followed by feature engineering. You want to convert the polymer structure into a machine-readable format. The machine or the computer knows what polymer is and how to represent it correctly. And then you can use your regression machine learning model to predict the properties. Like I mentioned earlier, we can predict the water permeability, salt rejection, ion selectivity, so on and so forth. And most importantly, if you want to discover a completely new materials to achieve some desired property, the generative machine learning will help. So for example, you can use generative AI to pop up the right polymer structures, give you the target permeability selectivity performance. Okay, so this is the whole framework we have been developing in the past 5 years. For sake of time, I just want to talk about the first 3 steps, how we start from a polymer repository, do the feature engineering, so we can establish the machine learning model to predict the water membrane's permeability as well as the ion rejection. Okay, so they will guide the experimentalist to specifically tailor the membrane for the different purpose. But before that, I want to highlight the current issue we are facing in this particular field. This is a database or the paper people published about a couple years ago. They call the open membrane database. The purpose is great. So people collect in the history, what kind of the polymer membranes has been synthesized and the water permeability, that is the A value, and versus the salt permeability constant, which is normally considered as the salt rejection, as the B value, has been collected so far, okay? Unfortunately, the data is very noisy and is very bad. So we cannot rely on this existing literature data to train the machine learning model to do the prediction. So this is one of the major issue in this particular field. So to resolve this issue, so what we propose through NAUI and in collaboration with Jeff McCutcheon's group at UConn as well as Yuepeng at Argonne, so that is, we consider the potential molecular compositions, like I mentioned earlier, the different amine recipes we can use. And we computationally generated the polyamide membrane, like what I show you in the first part. And we are able to run these molecular simulations, thousands of them, actually we did already. So we can pretty reasonably predict the water permeability, the salt rejection or their partition from this high throughput molecular simulation. Then we are able to train a machine learning model to have pretty reasonable prediction regarding the different membranes chemistry or the structure, and we can use machine learning model, predict their water permeability as well as the salt rejection. That will guide the experimentalist by using the electrospray-based printing to manufacture this membrane, and we can test them and feedback to our molecular simulation as well as machine learning model. So this is the whole idea. And the reason why we want to use molecular dynamics is MD simulation is 100% under control. So we have everything streamlined. It's very detailed and it's very self-consistent. So this will intentionally avoid, the bias, the initial noise from your training data. Otherwise, your machine learning model is already biased. So there's no way you can do a good prediction later on. So far, we totally simulated over 1,000 different amine monomers, and we use a typical 90% cross-linking degree. We apply the feed pressure about 1.5 kilobar, and with the room temperature, and the total simulation time is around 150 microsecond, okay, for the 1,000 membrane we have been simulated. And we are able to extract the A value as well as the B value. Again, A value is the water permeability, B value is the salt rejection. And here are some examples of the amine monomer we consider, and they are all commercial valuable, so you can order them right away from Sigma Orange or many other commercial vendors. So we consider some small aromatic amines, similarly like the MPD monomer, some small fluorinated amine molecules, so this is particularly interesting. We know PFAS is not favorable, but we still introduce some fluorine atoms into the amine group. And we consider some cyclical aliphatic amines and the linear diamines, and some very large aromatic diamines along with some large triamine or tetraamines. So they have more amine functional group than diamine molecule. And regarding their topological diversity, as you can see, so they have variety of the aromatic rings from 0 ring structure up to 11 ring structure, okay, just a few of them, and the majority of it is in between. They also eventually have different cross-linking degrees, so they are not really fully cross-linked, similarly as experiments. So they have variety of the oxygen over nitrogen ratio from our MD simulation, and as well as the atomic diversity. So the amine monomers contain variety of the nitrogen atom. Smallest, like diamine, has 2, and some largest have up to 8. Okay, so very diverse chemical structure of the polyimide membrane we can simulate. And then we look at the A and the B value we are able to extract from molecular dynamics simulation. All right, so what we get is experimentally, so typically cover this red dots region, while our MD simulation results cover actually a little bit different region, so by these blue dots. So the reason why this is happening because experimentally people always use the MPD TMC classical recipe, so that's been given already, and it's from the open membrane database for this membrane. And what our MD simulation simulate is we simulate a very thin membrane, about 10 nanometer, which is actually realistic. So the thinner of the membrane, the higher water permeability you will get, which we see here. And we also simulate a variety of the amine chemistry, like I mentioned. So we consider some of the amine molecules never been imagined before by the experimentalist. So that's the reason why we can have some high water permeability and also different, salt rejections from our AMD simulation, compared with the existing experimental data. And then we can start training the machine learning model to try to predict this membrane's water permeability, is the A value as well as the B value. So we consider 2 different mode or the models. So the first one is we consider by using the Morgan fingerprint representing the fragment or the structures of the monomer, as well as the dry membrane's fractional free volume, the average pore size, blah, blah. And then we train a multi-layer neural network, okay, it's a dense layer, and trying to predict the water permeability as well as the salt rejection, the B value. And so this is fully based on the 1,000 MD simulation results. And here are what we get. So as you can see, so the B value, the salt rejection, the training testing was not so good. Water permeability, just a little bit better. So then we change our strategy. So we consider by using the chemical signature of the membrane along with the dry membrane feature. So these are the dry membrane, not hydrated membrane. And their fractional free volume, average pore diameter, their standard deviation, we try to predict the hydrated membrane properties, like their fractional free volume, average pore size, standard deviation, along with the water permeability and the salt rejection, okay? So after we change this, as you can see, so the salt rejection as well as the water permeability prediction can be significantly improved from our machine learning model. So this gives us the confidence the microstructure or the fractional free volume of the dry membrane, okay, is really play a very important role, which already mentioned in the first part, because the water going through the membrane through these channels, okay, or through these pores. And to further quantify that, we also use the sharp analysis to do the explainable machine learning. So regarding the water permeability, so the most important factor we found is actually the dry membrane's fractional free volume. So the water permeability increased with the average fractional free volume along with the pore radius. The chemistry are not that important because it's indirectly related through the fractional free volume as the pore property. And also we identify some of the aliphatic and the aromatic carbon, they actually reduce the water permeability, while the fluorine atoms with the sulfur and the double bonded oxygen increase the water permeability dramatically. So this is mostly likely because they reduce the membrane's wall friction with the water molecule. So that's what we believe. And if you look at the salt rejection, so the B value, so the similar thing has been observed. So the B value actually increased the salt permeability, increased the fractional free volume of the membrane again, along with the pore size. And the presence of the nitrogen, chlorine, and the aromatic carbon, they actually increase the salt permeability as well. Well, the introducing these aliphatic carbons actually reduce the B value quite a lot. And we also tested some other architectures from during this machine learning study. For example, I mentioned the dense multi-layer, neural networks we did. We also consider some other features like the molecular descriptors from Avadesk actually will reduce the performance of the model. We also consider some convolution neural networks or gradient-based, machine learning model. So they are not very good. So overall, the feed-forward neural network is the best architecture we found so far, to train this machine learning model, give the good prediction regarding the water permeability and the salt rejection. And here are some key observations for sake of time. I don't want to repeat them again, but hopefully I showcase we are able to, using the high throughput molecular simulation, generate good amount of data regarding the different membrane chemistry, structure, their water permeability, salt rejection. Then you can train the machine learning model as a surrogate model, which can make the predictions on the new membrane you never simulated before. Okay, so this is the rationale. So eventually we will provide the recipe to the experimentalist to decide what kind of the amine molecule you want to use, since this is polyamide membrane. So you will be able specifically tailor their water permeability, salt rejection, ion selection, so on and so forth. So overall, so we can explore over 50 monomers easily per day in the current stage. And towards the end, I just want to briefly highlight there's a knowledge gap and the research needs in the field. So again, so here are some of the important questions we believe from a simulation point of view. So the pore connectivity, their membrane structure, heterogeneity are so important to understand, the transport pathway. And the insufficient of the current molecular simulation validation, as most of people, they just take it as it is, but we know that is not the real case. In particular, if you consider neutron scattering for the water structures within the membrane, some in situ measurement techniques, there's also quite a lot incomplete understanding on the ion dehydration and the transport mechanism. If you want to do the lithium extraction or some critical minerals extraction, so this is particularly important. The force field, as I mentioned already, so it's playing a very important role, as well as some other external factors, because you have the different pH, temperature, electro-osmotic conditions, how they will influence the transport. That's another important direction. So that's why back to earlier in the year, so we had a very interesting symposium, in Paris. It's called Interdisciplinary Perspective on the Transport Mechanism in the Membranes and the Nanopores. So we have quite a few world-leading experts, Professor Emmanuel Malik, Pedro, and Chilin, and as well many others get together. So we had a 2-day fully discussion. So we wrote a perspective, and we just got a review back and revise that. So you will be online pretty soon. So we detailed, a very clear roadmap how we can, target or tackle this problem from a multi-scale point of view, from the molecular scale, microscopic scale, until the continuum scale. What kind of the theoretical model, computational model, as well as experimental technique we can leverage. So it's all detailed in this perspective. So stay tuned. So once online, I can share with everyone. And towards the end, I just want to highlight again, so the importance of this reverse osmosis, as well as the water transport, mechanism. So there's a very interesting, article is writing about our work. So they talk about everyone was wrong about reverse osmosis until now. So that's highlight our simulation to show how the water actually going through a polymer membrane to make the desalination or reverse osmosis more efficient. And of course, I want to thank my experimental collaborators, Manny's group, his former postdoc Li Wang and current postdoc Han Qing, along three of my postdocs here. All right. And thank you very much for your attention. So I'm delighted to answer any questions you may have.
Excellent. Thank you, Dr. Lee, so much. Yeah, if anyone has any questions, please feel free to drop them in the Q&A box. We already have some in here, so we can get started with that. Someone has asked, how does the machine learning model know if a polymer structure that it comes up with through generative, automation, how does it know that that's possible to manufacture?
Yeah.
And how is that like incorporated into your process?
Yeah, that's a great point. So as I mentioned earlier, so when we build our polyamide membrane library, so we use all the existing amine molecules. So they are all commercially available. So you can imagine it's like playing the Lego. So I have A component, I have a B component. My A component is amine molecule, which are all commercially available. My B component, I want to fix it as a TMC molecule, right? Following the traditional polyamide membrane. So our machine learning model actually will take these two components together and to extract the most important chemical features and do the prediction. So when you come back, you want to identify what is the next best candidate, right? So you already start from the existing known molecule. You don't start from something do not exist yet. So this will elevate the manufacturing issue or the synthesis issue. Yeah, we can design some molecular structures are really novel, but probably none of the chemists will be able to make it. So that will be the big issue, right?
Great. Great, thank you so much. Um, and then could you talk about like how or if these reverse osmosis models are being used now?
Yeah, so these reverse osmosis models are mostly used to do the process optimization because the membrane I highlighted so far is only like one stage for the water treatment, right? Or pure water purification. So in reality, so this is the real water plant. And if you really go in there and you visit there, you will see it's actually thousands of the modules they work together. So you have a multi-stage reverse osmosis. They collectively work together for the whole, desalination or the water treatment. So typically, so these transport models, so they will be used embedded during this process modeling because you need to know, right? So my wastewater, they going through this particular module and what kind of the water flux or the other species and will be able to get, and then how to move on to the next stage, so on and so forth. So that's basically being dramatically used for the process control and the process optimization
Hmm,
for the whole desalination plant. Yeah. So I
got it.
hope I addressed the question.
Yeah, great. Thank you so much. And then this is more of a question about like the water chemistry and
Yeah.
how, like you mentioned, like pre-treatment or like where this happens along the treatment train, how that
Right.
influences membrane like efficacy.
Yeah.
So is the permeability or like have you been able to see if the permeability of the membranes are improved by like an increase in dissolved oxygen, for example?
This is a great point. So this is also one of the limitation of the molecular dynamics simulation because we cannot really simulate the real water chemistry because just for example, the seawater alone, they are not just having sodium chlorine, they have many other, ions as well as organic contaminant like a boron or some others. But because of limitation of the molecular simulation, the time and also the computational cost, so we cannot put in every species into the molecular simulation model. So we are not able to really, quantify how each different species will, influence the water permeability, their performance. But if we know there are some factors that are important, for example, we know borons is one of the big issue when you want to do the RO or desalination. So we actually can place the boron molecule into the feed, and we can simulate how the boron molecule going through the membrane along with the water or the sodium chlorine ions together, so we can quantify that. But so
Interesting.
far we haven't, yeah, because the computational cost and also the limitation of the molecular model.
Yeah, I imagine, especially when you're dealing with complex waste streams, it can almost turn into
That's
like a
right. Yeah, that's right. Yeah.
black box model where you
For
like
single species, that can be easily done, but when you have a multiple species, in particular, very complicated, that will takes forever for simulation to do. Still, experimentally, where experiments will play a very important role for this. Yeah.
Awesome. Thank you. Um, and just based off time, we just have a couple more minutes left. So, I wanted to thank you, Dr. Li, for your time. This was a really great presentation. Really digging deep into this work. And then thank you also to the folks who are online for your participation, for all the questions. This was great. If you're interested in continuing to listen into these Third Coast Water Seminar series, they do occur monthly. So our next one is going to be on November 5th at 2 p.m. It will be virtual, and it will be focused on the work from Dr. Reshmina William, who is going to be talking about the water sector innovation, de-risking, piloting, and trials. Also, if you're interested, this presentation and seminar will be posted on our YouTube channel if you would like to rewatch or share out with your friends. And with that, yep, thank you everyone for your time. This was great.
Yeah. Thank you. Thank you so much.
Thank you.